Assalamu alaikum. Our lecture today on biochemistry of amino acids and protein, structure and function. First object, definition of amino. First definition of protein. Protein is a polymer formed of subunit called amino acids linked together by peptide bond. The term of protein is applied to describe molecule more than 50 amino acids. Less than 50 amino acid is called polypeptides. Second object, structure, classification of amino acid. Amino acid, although about 300 amino acids exist in nature, only 20 of them are present in human to form protein structure. All amino acids are aliphatic and L amino acid. L amino acid means that the amino group are present in the left side of the first carbon. Classification of amino acids. Amino acids are classified chemically into neutral amino acid, basic amino acid, and acidic amino acid. Aliphatic neutral amino acids are composed from glycine, alanine, valine, leucine, and isoleucine. Each amino acid of them has specific function. Glycine is used for the detoxification of benzene, synthesis of protein, hemoglobin, bile salt, purine, reduced glutathione, creatine, serine, and choline. Alanine for glucose synthesis, valine, leucine, and isoleucine for acetyl coenzyme A, succinyl coenzyme A, and propionyl coenzyme A. Aromatic amino acid, which are phenylalanine and tyrosine. Both of them are used for synthesis of thyroid hormone, adrenaline, noradrenaline, melanin pigment, and cortisol and phenol. Hydroxy amino acid, for example, serine and threonine. Serine are used for synthesis of choline, sphingosine, cephalin, and cysteine. Threonine also is used for synthesis of intracellular protein glycosylation and phosphorylation, synthesis of acetyl coenzyme A and succinyl coenzyme A. Sulfur-containing amino acids, cysteine, cysteine, and methionine. All of them are used for synthesis of S-adenosine which is an active form of methionine and is the main methyl donor in our body. Synthesis of pyruvate and glutathione, detoxification of hydrocarbon. Heterocyclic amino acids, for example, proline, hydroxyproline, tryptophan, and histidine. Proline and hydroxyproline are used for synthesis of collagen. Tryptophan used for synthesis of nicotinic acid, melatonin, serotonin, ketone bodies, indole, and ketone. Histidine are used for synthesis of histamine, which is an inflammatory mediator, synthesis of carnosine and anthirine, also for synthesis of ergothioneine, which is an antioxidant. Acidic amino acid, for example, aspartic, asparagine, glutamic, and glutamine. Aspartic acid and asparagine used for synthesis of asparagine is important for protein cross-linking and glucosylation, synthesis of purine, synthesis of pyrimidine. Glutamic acid and glutamine used for synthesis of removing ammonia from the brain, synthesis of gamma-aminobutyric acid, which is a neurotransmitter, activation of folic acid, urea synthesis, synthesis of glutathione, and synthesis of asparagine. Basic amino acid consists of 5 amino acids: ornithine, arginine, citrulline, lysine, and hydroxylysine. This group of amino acids contain more than one amino group. Ornithine are used for sense of citrulline, intoxication of aromatic amino acid, sense of polyamine for cell cycle regulation, sense of glutamic acid. Arginine used for sense of creatine, sense of urea, arginine phosphate, sense of nitric oxide. Lysine activated biotin in decarboxylation reaction, sense of collagen, and sense of carnitine, which is vitamin T. Second classification of amino acid according to metabolic base. Amino acid can be classified into ketogenic. This type of amino acid can sense ketone body, cetone, acetoacetic acid, and beta-hydroxybutyric acid. This type of ketogenic amino acid include leucine, which is pure ketogenic amino acid. Other type of amino acid that can sense ketone body and glucose called mixed or ketogenic and glucogenic amino acid. For example, lysine, isoleucine, thyroidine, tryptophan, and phenylalanine. The remaining amino acids are pure glucogenic. The third type of classification of amino acids according to biological and nutritional value into essential amino acid and indispensable amino acid. This type of amino acid cannot be synthesized in the human body, should be taken in diet, have a main role in both growth and health. Deficiency of these types of amino acids lead to nutrition deficiency disease. This type include valine, isoleucine, threonine, tryptophan, arginine, leucine, lysine, methionine, phenylalanine, and histidine. Arginine and histidine are semi-essential amino acids because it will be both are synthesized in the body, but its level is are not sufficient for the requirement of our body. Non-essential amino acid, dispensable amino acid. This type of amino acid can be synthesized in the human body, may be taken in the diet, and have a less role in health and growth. Their deficiency does not lead to nutrition deficiency disease. The last object describes the structure and organization of protein. Protein structure are classified into primary structure, which are linear form of amino acid held together by peptide bond. It has two terminal sites, C terminal end and N terminal. C terminal is a carboxylic free end and N terminal is amino free end. No liable for denaturation. Second structure is a folding of polypeptide chain to coiled structure. The bond present in this type is peptide, hydrogen, ionic, and disulfide bonds. This type is liable for denaturation. The third type of protein structure is tertiary structure. This type is three-dimensional, more complicated folding and superfolding of second structure polypeptide chain due to interaction of amino acids located far apart. It may be globular, for example, myoglobin, or fibrous, for example, keratin, collagen, and elastin. It contains all types of protein bond, so it's liable for denaturation. Last type of protein structure is quaternary protein structure. It consists of two or more polypeptide chains connected by other than peptide bond. If there is two subunits of protein, it's called dimeric. Three subunits is called trimeric. and is formed of several subunits called multimeric. It contains all types of protein bond, so it's liable for denaturation. For example, of quaternary protein structure, it's a common type of protein structure. It are globulin in hemoglobin, lactic dehydrogenase, apoferritin, keratin kinase.